Very good afternoon, everyone. Myself, Dr. Akanksha from Biosensors University. Today, we'll be talking about biosensor. Biosensor basically is a short form for biological sensor. It's an analytical device which is used for detection of any analyte that combines a biological component and a physiochemical detector. Professor Lyndon C. Clark is termed as the father of biosensor. Biosensor basically incorporates a biological sensing element either intimately connected or integrated with a transducer. Basically, recognition in biological sensor or biosensor is based on the affinity between complementary structures such as enzyme and substrate, antibody antigen, receptor or hormones. Selectivity and specificity basically depends on biological recognition of systems that are connected to the suitable transducer. So basically, it converts your chemical response into an electrical signal where it gets detected, recorded, and the information gets transmitted regarding any physiological change or chemical reaction occurring in a device. It determines the presence and concentration of specific analyte or a substance in a test solution. So main features of biosensors basically include stability, sensitivity, reproducibility, and most importantly, it should be cost effective. So talking of basic principles of biosensor, the basic principle of biosensor basically involves three elements. First is biological recognition element, which is highly specific towards the biological material analyte produced. Second is transducer element that detects and transduces your chemical signal, or you can say signal from biological target, where it is detected by a receptor molecule and is converted to electrical signal due to the reaction. And third, after transduction signal from biological component to electrical signal, where sometimes amplification is necessary, it readouts the reading after processing the values, and the values are displayed at the end with the units. Now, components of biosensors, as I told, starts with biological recognition element, which can comprise of your analyte and bioreceptor. Now here, if you see, this is the analyte which is shown here in different colors, and this is the bioreceptor. Be it, suppose these analytes are substrate that has to bind to specific enzyme. Now it will only bind to that specific enzyme based on, as you have studied lock and key model. If it is of particular shape, it will go and bind to that particular bioreceptor. So once the analyte is bounded to the bioreceptor, transducer will read that signal and convert this chemical response into electrical signals, which will be detected by your detector. So here, transducer can be converting your pH change, speed change, light change into your detectable measurable signals, say photon counter or piezoelectric device will record this reading. So these are the major components of your biosensor. So as I told, first component is a biological element, the component which is used to bind to the receptor or the target molecule, which should be highly specific and stable under storage conditions. Now, many substances can be used as biological element, but most probably microorganism, tissue culture, enzyme, nucleic acids, or plant proteins can be used as a biological element. Next, why these biological elements are required or what are the functions of this biological element. So basically this interacts with the target compound, reacts and gets detected. Second, it should be capable of detecting the presence of the target compound in this test solution. And the ability of bioelement has to react with the target compound. That is the main basis of your biosensor. Then only that chemical reaction will be recorded by a transducer and converted into electrical signals. So second component is your physicochemical transducer, which basically acts as an interface or connection where it measures your physical change that occurs after the reaction at the bioreceptor and then transforms it to electrical signal. Third and most important component is your detector. So signal once given from the transducer are passed to the microprocessor where they get amplified, analyzed, and then data is collected, converted into their particular concentration units, and then displayed at the device, which can also be stored for a later purpose. Next, principles of detection. So here, as I told, detection will be done after transducer has given a signal. It can also measure the change in mass, electric distribution, light intensity, or say heat, which can be detected by piezoelectric device, electromechanical device, optical or calorimetric device. So here is the block diagram that includes segment, as I told, sensor, transducer, and signal processor. Again, here, if you see, analyte has to bind to the sensor. So in the first segment, sensor is the responsive biological part, where second segment is detector part that will analyze or detect the chemical reaction which is going on and convert those signal with the help of transducer, and it will display in the accessible way. The final section is comprising of amplifier, which is known as signal conducting circuit, where it will take this signal, amplify it, and signal processor at the end it will display. So basic principle, as I told, includes immobilization of biological material, and a contact is made between this immobilized biological material and the receptor, or you say transducer. Now the analyte, once it binds to the biological material, it forms a complex or a bound analyte, which in turn produces electronic response, which is detected or measured using your detector. Sometimes analyte is converted to a product which could be associated with release of heat, gas, or electrons. Now, this transducer can convert the product into electrical signals, which can be amplified for data purpose. So working principle, if you see, analyte diffuses from the solution to the surface of the biosensor, where analyte reacts specifically and efficiently with the biological component of biosensor, and then this reaction changes the physicochemical properties at the transducer surface, where transducer reads it. Now this leads to the change in the optical electronic properties of your transducer surface. So the changes in optical electronic properties is measured by the transducer, and is converted into electrical signals, which later gets detected by detector. So usually a specific enzyme or biological material is deactivated by some of the usual methods, and the deactivated biological material is again brought into the contact of transducer. So the analyte connects the biological object to a shaped clear analyte, which in turn gives you electronic reaction, and that is recorded by the detector. Clear? So here, if you see again a schematic biosensor diagram, you have a bioelement, transducer. Now, bioelement, once the chemical reactions are done, chemical signals are sent to the transducer, which converts it into electronic signals. Now, electronic signals are taken by amplifier, gets amplified and signaled to the processor. At the end, a unit, which is given by a processor, will be displayed with units. Now, how does they work? Basically, it involves a quantitative analysis of various substances by converting their biological actions to measurable signals. Now, generally, the performance of biosensor is mostly dependent on specificity and sensitivity of the biological reaction which is occurring, and stability of that particular enzyme which you are reacting with a particular substrate. So an ideal biosensor is known to have following properties. First thing, the output signal must be relevant to the measurement environment. Second, the functional surface must be compatible with the transducer. High specificity and sensitivity is required. Sufficient sensitivity and resolution is required. And fourth, and the most important thing is sufficient speed of response and sufficient dynamic range is required. So insensitivity to environmental interference should be compensated. Clear? So basic characteristics overall, what a biosensor should have first is linearity. When you talk of linearity, it should be high detection for high substrate concentration, and it should always be able to detect minute concentrations also. So linearity here means with higher doses, the reading should increase. With lower doses, it should show decrease in the reading. Sensitivity at the same time shows the value of electrode response per substrate concentration. It should be able to read the minute readings also and highest readings also. Then comes selectivity. So chemical interference must be minimized for obtaining the correct result. That means whatever bioanalyte you want to test should only get detected rather than other elements which might also be present in the environment. Then response time is equally important. The time necessary for this is more than 95% response. Okay, it's not that you keep biosensor, some of the time it gives response, the other time it does not. So it should give response for maximum 95, or say not maximum, but minimum 95% of response should be recorded. And again, speed of response, it should not take a longer time to give you result. So the typical sensing techniques used nowadays are fluorescence, DNA microarray, plasma resistance, impedance spectroscopy, scanning probe microscopy, sometimes Raman spectroscopy can also be used. So here we'll be discussing different types of detectors. First, we'll start with calorimetric, where it detects change in the heat. Optical biosensor, resonant biosensor, piezoelectric, ion sensitive, electrochemical detectors or biosensors. Electrochemical biosensors can also be termed as conductimetric sensor, amperometric sensor, and potentiometric, depending on what it senses. So here, if you see talk of calorimetric or thermal detection biosensor, it basically uses absorption or production of heat, or you can say it measures the changes in the heat production or absorption. So here, total heat produced or absorbed is noted as alpha molar enthalpy or total number of molecules in the run, and temperature is sometimes measured by enzyme thermometers. Advantage of using that, no need of frequent calibration is required for calorimetric detector, and it is intensive to optical and electrochemical properties of the sample. Now, where it can be used for detection of pesticides, pathogenic bacteria, these type of biosensors can be used. Talking of ion sensitive biosensors, so surface electrical potential changes when ion and semiconductor interacts. So this measures change or change in electrical potential can be detected by ion sensitive biosensors. Basically, detection of pH can be done using this. Electrochemical biosensor, if we talk of, so chemical grants or produce or consume sometimes ion, which will lead to production of electricity by movement of electrons. So these electrical properties of the solution can be measured using electrochemical biosensor, which again, as I told, can be conductometric, amperometric, or potentiometric. Now, this type of detection can be used for hybridized DNA, DNA binding drugs, or sometimes glucose concentration monitoring. Optical biosensor, as I told, is for colorimetric for color, which measures the change in the light absorption, photometric for light intensity, and again resonant biosensors takes the change in waves or you say resonant frequencies. Piezoelectric biosensors usually is to detect a specific angle at which electrons are emitted or absorbed in the substance exposed to the laser light, like gold, quartz. When vibrations are there, under this influence, it detects your frequency or changes frequency can be checked. Sometimes change in mass also can be checked by piezoelectric biosensor. So conductimetric biosensor measures basically electrical conduction where double layer charging per processes of concentration polarization can be read. Amperometric is high sensitivity biosensor where it detects the electroactive species present in the biological test and also parameter that it measures is current. As current is taken in ampere, so we term it as amperometric biosensors. Next, we have potentiometric biosensors. Potentiometric, as term suggests, potential difference when voltage is applied to an electrode, solution current flows, because of which potential difference is observed. Be it any electrochemical reactions, that oxidation reduction potential can be measured using potentiometric sensors. So application wise, why we need to read this biosensor is it can be applicable for food analysis, study of biomolecular interaction, drug development, crime detection, medical diagnosis, environmental field monitoring, quality control, and even biological warfare agents can be used. And most importantly, pharmaceutical industries are working for making different biosensors for detection of several adulterants also. So talking of applications in medicines and health, biosensors are successfully used in quantitative estimation of several important substances in body, say glucometer for glucose, cholesterol, urea. Glucose biosensor are used for diabetic patients as well for regular monitoring of blood glucose. Blood gas monitoring can also be done like you check your partial pressure of oxygen, carbon dioxide during critical care or surgical monitoring. That can also be done. That is also again a biosensor. So every drug can be tested using a biosensor or toxic compound produced in our body can be detected using biosensor if you talk of medicine and health. Next, we talk of applications in food industry. So here, it can be used for monitoring fermentation products. It can be used to check the freshness of fruits, vegetables, or say stored fish, where stored fish can be detected by ATPase. ATP is not found in spoiled fish. So this can be detected using ATPase enzyme. A biosensor can incorporate ATPase enzyme and can be tested. Likewise, can be used for application pollution control, environmental monitoring, where biosensors are helpful to monitor environmental change and pollution. Or this can also detect the concentration of pesticides, which can again be monitored by biosensor. So it has a wide application, be it environmental detection of, say, measurement of control of water, soil, or air, or any contamination. So determination of pesticide usually is done by potentiometric biosensor. And the most important application is military. In military, where biosensors are developed to detect the toxic gases and other chemical agents used during the war. DNA biosensors is also used to detect your criminals also. So this was a general glimpse of biosensor, where we were talking of principle, components, and applications of biosensors. Thank you and have a nice day.